Great. Hi, welcome everyone to Nucleus community meeting, the last of the year. Um, and we're very excited to hear from Anton Molina today. Um, Anton is the director of open source ecosystem at bNext. This is the open core synthetic cell company based in San Francisco in the US. Anton completed his PhD in material science and engineering at Stanford University with Professor Manu Prakash, and his work brings together material engineering and open innovation practices to develop transformational technologies. Previous projects he's been involved with include processing agave fibers into absorbent materials for use in menstrual hygiene, hacking cotton canning machines to produce air filtration materials for pandemic preparedness, and studying physics of evaporation to understand the dynamics of 2D crystalline materials. And today he's with us to tell us a bit more about what's new and what's next with his company. So with that, I will pass the baton over to you, Anton, to tell us a bit more. Thank you and welcome.
All right, thanks for the introduction. Share my screen and look for my slides. Let's see. Oh God, started at the end. Okay, right. So thanks again for the introduction. So as Jan had mentioned, I'm Anton Molina. I'm the director of open ecosystem at bNext. And I want to start the talk just with this notion of collaboration. So this has kind of been an inspirational image, I think, throughout our process of building bNext and thinking about biology and open source and how that all ties together. So what you're looking at is an image of the collaborative development of the Git software project, which is foundational infrastructure for a lot of modern software engineering projects. And the first thing that comes out is that each one of these dots represents a contributor, right? Somebody making a contribution to either the code base, the documentation base, or any of the other number of components that it takes to make an open source project successful. And the first thing is just the scale of it, right? There's hundreds of people that are collaboratively building this thing. And looking across the field to biology, right, the picture is a little bit different, where most biological collaborations, if they happen at all, are often pairwise and frequently require somebody physically being co-located in one laboratory or another to actually work together and build on the same system. And so one of the questions is why that's the case. And at least part of the answer comes down to the systems that we're trying to engineer. So in this table, right, these are sort of the arguably the three most well-studied organisms, the industrial workhorses of biotechnology. And it remains that most of their genome is unknown. And underneath that, right, underneath that sort of unknown complexity, that unknown sort of genome, right, lies an enormous amount of complexity that gets manifested either in terms of in this David Goodsell imagery of the spatial complexity associated with how a cell is organized internally, or complexity in terms of its metabolic network. So on the right there is a depiction of the metabolome of I believe, E. coli. And both of those are pushing the boundaries of scientific understanding, and as a result, right, it makes for kind of poor substrate for actual engineering. And so you end up with this framework that despite the title, right, we're still sort of researching biology and not necessarily engineering it. And so what's an alternative approach to working with biological systems, right, is this idea of compositional biology, and this comes a lot from the synthetic cell community, specifically the bottom-up synthetic cell community, where the idea is to build up systems that have the sort of properties and behavior of living cell-like systems, right? But they're built from known components. And so in this case, this is sort of an animation of what we mean by compositional biology, where we're literally mixing the building blocks of living systems from ribosomes, proteins, small molecules, DNA, and assembling them into cell-like objects that are admittedly quite simple. But because we can keep track of all the components, we know the structure inside, right? It affords us a couple other tools that we get along with this, which is the digital description of synthetic cells, the complete description of its genome, right? We can put an entirely synthetic genome into this. And so taken together, it points towards the ability to do things like forward design of these systems. And so being able to imagine, CAD/CAM technology for biology, and predictive modeling. And so the enabling technology under this is what's known as the pure system. It's a type of cell-free expression system that's made from purified components, which stands in contrast to lysates, right? So rather than just cracking open the cell and making use of the transcription translation machinery there, we're individually purifying each of the components of the transcription translation machinery, right? And so this schematic roughly corresponds to all of the processes that underlie the transcription translation factor. And when you look at the literature, there's a lot of demonstrations that extend the capacity beyond this sort of standard transcription translation, right? There's papers out there that show the ability to increase the longevity of the system by introducing additional energy regeneration sort of subsystems. There's things that expand the number of proteins that can be expressed through the introduction of chaperones, either at the membrane or in the cytosol. There's functional pores that allow for selective transport of molecules into and out of the system, right? There's synthetic components like RNA logic circuits that allow you to do logic in the cell. The problem is that each of those are sort of one-off demonstrations that are done under slightly different circumstances. And there's a missing sort of framework and incentives to bring all of those components together. And what's sort of tantalizing and inspirational is imagining what could happen if those components are brought to cooperate with each other and can interoperate with each other, right? It would be potentially a powerful framework for bringing together engineered biological components into a system that we can design, model, and engineer in a way that looks like engineering much more so than research, right? And so to cross this, what we call the integration gap, right, we need some framework for collaboration. And so we've been spending a lot of time prototyping and building what such a framework would look like. We call it Nucleus, and it's fundamentally an open source ecosystem. So what is an open source ecosystem? It's a organization that enables the ongoing asynchronous development of an open source product that's designed to be publicly accessible, modifiable, and distributable by anyone under an open source licensing model. So that's a definition that comes more or less unmodified from the NSF, and I think it's a quite good description. And so with Nucleus, the open source product is it represents quite a few components. We call it the Nucleus distribution, right? And so it refers to the set of things that are needed to make synthetic cells. So that includes designs, it includes the plasmids, it includes the protocols for taking the plasmids and making them into cells, and all of the sort of supporting tools needed to do that. And on the right is sort of if those are the nouns, right, on the right is the verb, right? The collaboration framework, the set of processes that we use to iteratively develop this collection of artifacts, the protocols, the materials, and so on, needed to make increasingly performant synthetic cells. And so I'll spend a little bit of time just how we kind of built this project from the start. So about 2 years ago, 2.5 years ago, BNext received its first contract to develop uh, an open framework for building synthetic cells, and that was framed in terms of a classic sort of toy integration problem, right? How can we integrate two functional synthetic cells to make a more functional synthetic cell? And so in this case, we have a detector cell, which is able to detect ATC and emit a reporter protein. We have the responder cell, which is able to take two small molecule substrates and produce a quorum sensing molecule, IVHSL. And then finally, the integrated system, which is able to go from ATC to that quorum sensing molecule and then interface with engineered E. coli that emit GFP in response to the quorum sensing molecule. So that was sort of the engineering challenge. And the goal was, not to do this and write a paper and be done with it, but to think about the sort of holistic framework needed to make future integration efforts easy and extensible, right? How can we create the structures needed to bring in more integrated components? And so we broke it down roughly into these elements, right? We needed a framework for designs. We needed certain tools that could be shared and interoperable, protocols, documentation, and sort of the abstract classes of things that needed to be integrated, right? Thinking about the sort of membrane as distinct from the cytosol, as distinct from the integration of those two. And then the design philosophy was always to be sort of working in public and iterating regularly. So that's where we get roughly this 3-month cycle of regular releases. And I think we've mostly held to that over the last 2.5 years. And to where we are now, we've continuously developed that process and as we think more about what it takes to create a self-sustaining, organization that has a lot of different external contributors, we've had to add more complexity to the process. How do we think about external contributions, right? The previous work was done mostly within one lab and building the core processes, but going forward, we want to be able to create a robust framework so that external contributions can be made from a broader developer community. And how do we think about maintaining quality of those contributions as we go on? How do we create ongoing dialogue with the community? And so we've ended up with this framework that we call the Nucleus development cycle. And in a sort of in the second half of the talk, I'll go into each of these components in turn. But before going there, I just want to sort of paint a picture of a look ahead, right? So we started with our first integration challenge, and then received support to basically build a type of foundational cell that we call a developer cell, which brings in core capacities into the synthetic cell. So this is the idea of taking pure and then making it better for a synthetic cell context. So adding things like a better energy regeneration module, adding a membrane insertion protein, the SEQI-AG system, adding a protein degradation, right, to be able to turn on and off expression. And then, so that's on the cell side. And then on the sort of community side, we've recently launched a project called the Developer Cells Project, which is rather than building the complexity of the synthetic cell itself, it's about bringing in more developers to sort of test and develop the framework itself. And so there we have this sort of node or team-based structure where there's 3 nodes, 2 in addition to bNext, which is serving as sort of an integration partner in that collaboration, are using this framework to develop 2 demonstration projects. And the idea is really that sort of the patterns of collaboration that emerge from this effort, right, are things that can scale from 2 to 3 nodes to many more nodes, and turn the practice of synthetic cell engineering from something that is done through the accumulation of papers, which are not really designed for integration or interoperability, into a practice of building on a shared body of knowledge. What you see below is sort of a multi-year arc of what that might look like, right? So this is courtesy of Richard Murray at Caltech, um, uh, sort of presenting the challenge that if we want to get to synthetic cells of a certain level of complexity and a certain level of operational lifetime, it's going to require collaboration at many different scales. And so we're hoping that this framework can serve as the basis for that scaled collaboration. And so with that, I want to walk through roughly the components of the Nucleus development cycle just to give a sense of like what might be considered a user journey. It would start with the documentation. So you can imagine a scenario where you want to access or implement the developer cell or some other type of synthetic cell that is of interest to you. The idea is that you start at the documentation, you go have a look, and um, and through navigating, a website, clean presentation of information, you would have a protocol, you'd have pointers to find the necessary materials, how-to and tutorial guides um, to sort of run and execute experiments, and also just best practices for working with these systems, since there's a lot of sort of tacit knowledge. This video below kind of gives an example of what one of these sort of protocol or specification pages would look like. It has call outs for what are the critical materials that you should really think about, not just the name of the product, but like the actual SKU, because the one vendor has exactly the right thing. Links to design files for the relevant genetic components, and some anticipated behavior. So with protocol in hand, one can go into the lab and implement it, do an experiment, collect data. And then comes the question of how do I report back what I've done? Or better yet, right, if I've taken that sort of standard cell and modified it in some way, how do I not only report back that, hey, it works, right, and giving confidence to others within the community that the thing works, but share an improvement, right? And so that brings us to shared metrology and ultimately software tools. And so the set of software tools within Nucleus we call the Cell Development Kit. And it's a set of open source tools that are designed to basically offer sort of work out of the box functionality for cell-free and synthetic cell workflows. Right now, the emphasis is mostly on the analysis of experiments. And I'll highlight this kind of through the specific case of what is sort of a de facto standard in the cell-free expression community, which is just the GFP expression assay, right? It's a de facto standard, so it's something that people broadly do, but there's a lot of inconsistencies in how people actually perform the assay. So there was a NIST workshop that highlighted some of these gaps. So for instance, a lot of people don't present a full curve, they present just an endpoint, right? It's a bar graph, leaving a lot of information on the table. And that information would be useful, right? If we look a little bit further down the line, right? If I want to think about modeling these systems, it's important to have a full profile because the full kinetic trace is amenable to modeling. If I want to think about engineering time-sensitive systems, knowing that expression profile is also valuable. And then it's not just about sort of the mathematical analysis, it's also about the underlying materials, right? Often there's sort of discrepancies between people reporting the performance of their system using DGFP, SFGFP, EGFP, right? So harmonizing on a sort of standard reporter becomes valuable as we imagine building a collective base of knowledge, right? And so it just highlights one of the aspects of building a holistic framework, right? We're not just thinking about the software tools, but we're thinking about how the materials and the protocols tie together to create something useful. So this panel here, right, just shows some of our early implementations of this. I guess the other kind of fact buried in there is, if you want to extract the kinetic parameters, it's often not easy to do it if your tool of choice is Excel, for instance, right? It might be implemented inconsistently. And so basically having just an out-of-the-box toolkit that everyone kind of in this community is using facilitates shared measurement. And so that's roughly kind of working at the scale of the lab, at an individual lab. Over time, we want to expand it to more areas of the sort of research pipeline, right? So not just analysis, but also the design of the design and construction of experiments, the execution of experiments. We can think a little bit about, automation protocols that could then kind of create a continuous pipeline from design, execution, and analysis, and then ultimately modeling. And then given a sort of shared digital language for individual labs, right, it also opens up interesting possibilities for imagining how things get used across labs. Sorry. Um, Sorry. And then, right, so things like, um, uh, right, how can data be, FAIR compliant across the ecosystem? By having standardized analysis pipelines, right, you're able to embed consistent metadata. It opens up new ways for sharing the data through, things like developer notes, which I'll get into in the next few slides, and then other ways of reuse, right? Being able to sort of tie in the performance of a particular plasmid construct with its performance in cytosol, right, with reference to specific sort of compositions. It enables sort of just greater reuse and faith in the materials that we're building on and using as a community. And of course, this means we have a kind of a challenge, right? So as sort of the software tools increase in complexity, right? There's a concern around accessibility, and so this is roughly the challenge that Nucleus Hub aims to address. So Jupyter notebooks, I think from the earliest we knew were going to be part of the answer. And if you're not familiar, Jupyter notebooks are essentially computational documents that bring together code, equations, visualizations, narrative text into the same document. And what this means is that data analysis and narrative text are co-located, right? So your data is very well contextualized, which matters a lot for biological systems where there's a lot of just, a sentence can matter. And then on the right here is just roughly what that looks like if you haven't seen a Jupyter notebook before, right? I'm blending code, pulling in like the CDGA with analysis curves and pointers to the actual data sets. Of course, it still doesn't answer the question of how I share this. So this is where another kind of open source tool has proven very enabling for us, which is Jupyter Hub. We've taken Jupyter Hub and made use of its sort of open source affordances and modified it so that it works with some of the other tools that we're developing, such as developer notes, which I'll get into later. But what it enables is essentially every developer within this community can access essentially the same computer, right? They don't have to develop a software environment on their own. They don't have to maintain dependencies. You can just log in and make use of these tools. And also, it's a venue for sharing data as well, which can be kind of cumbersome to emailing CSV files back and forth and so on. And then a look ahead, being able to accommodate more challenging data sets like microscopy data sets, where access to computation can be a barrier, and there's far more variance in how people extract data from those data sets, right? And often, everyone has their own sort of pipeline for extracting data, which makes the interoperability a little bit of that data more challenging. And then this video roughly shows an example of the interface here with Jupyter Hub. We've modified it a little bit since then, but the rough idea is I'm accessing this interface from the browser, and I'm able to pull in plate map, data set, and then make use of some of these functions that just sort of generate, plotting functions with same defaults for presenting kinetic and sort of time series data. Um, uh, and then sort of within this environment are also things like standard plate maps and there's documentation for how to use that so that the metadata is roughly consistent, which makes it possible for somebody, say, in the modeling world that wants to know, they frequently want to know, what is the exact composition of your system, right? And what is the resulting output behavior? All that's presented in a sort of consistent way. So as more and more examples of this get sort of seeded into the system or into the sort of collective database, it's built from the start for usability from that perspective, right? And the other thing that we've done here is we've integrated Jupyter Hub with communication tools. So what you're seeing here is basically a mapping of a figure of an output from a Jupyter cell directly into a dev note, which is the next topic. So developer notes. Developer notes are essentially the answer to how does one contribute to the Nucleus ecosystem. And so it looks something quite a bit like like a paper, right? It's the standard way of communicating scientific knowledge. But given that we see it much more as an engineering tool, right? And it's community run, so we have the ability to create a space for things that often fall through the cracks of traditional publishing, right? So things like technical notes, things like negative results, things like I just ran one sort of set of parameters, but I think it's interesting. You can share it here and get feedback on that work, and sort of just announce to the world that this is something that you're working on, and ideally work with others to collaborate on that. And the other aspect of this, given that it's sort of again built on sort of open source Jupyter tools, it means that we can prioritize like certain types of research artifacts to be like first class citizens of a research article. So in this case, right, we're working on a way of just how do we better highlight, say, DNA sequence information directly into the publication. So for instance, here this is basically a seqviz widget that's part of the developer node. And so then the metadata, the file associated with that is now embedded in the article's metadata. And so you can imagine when there's multiple dev notes, right? It's much more easy to access. Okay, how many dev notes feature this particular sequence or have characterized this particular sequence? And because developer notes again are not just static PDFs, but entire sort of directories, right? Or you can think of them as like a paper repository, right? They contain the characterization data, they contain the experimental parameters all in one place. And so then, the paper kind of becomes really this a building block in a much larger sort of structure, right? Dev nodes talking to each other, the data talking to each other, and it's all sort of searchable and machine readable. And it's been really internally, right? We develop modules for ourselves, right? This is the PPK module, which is an energy regeneration module in Pure, and it's been really, I think, nice to kind of work in this flow where we set some milestones for ourselves, and then every time we hit a milestone, we get to push out that contribution, and it's I think from a sort of data management point of view, really valuable because we have an incentive to curate along the way, which makes it just really easy to sort of stay on top of things and also share it with others. to have tangible demonstrations at intermediate steps is a nice thing to have. And ultimately, dev notes will culminate in posting validated protocols on the documentation page, which sort of completes the arc, right? And then somebody can take that validated protocol and start the cycle all over again. So that's been a lot about the sort of digital infrastructure of Nucleus and sort of the developer journey to go from using a protocol to now contributing a new protocol. But we also do a lot of things in the physical world. So I'll give you a brief highlight here, right? So workshops has been sort of a very powerful sort of tool and I think valuable service for the community in terms of just bringing together people and introducing kind of this different way of working, and just getting to know all the different kind of points of view within this engineering community. And so this is a highlight of several workshops that we've run over the last year and a half, from making liposomes to making pure to sort of imagining how, what collaborative development looks like. And throughout each of these workshops, we've gotten increasingly better at sort of this practice of what might be called continuous science, which is, within a week, we design an experiment, analyze it, and then have it out as a developer note, right, within a scale of a week, which is quite quick, I think, for how most people think about working with biological systems. And then finally, and this is a very foundational component, is again supporting everything we do with materials, because having access to high quality, well documented materials is like absolutely essential for doing biology and compositional biology. From the earliest, we've supported open access plasmids that are available under the OpenMTA. These are the plasmids that would be used to generate all of the 36 component proteins in the pure system. And going into 2026, we're moving beyond not just supporting the plasmid collection, but also having a kit, which corresponds to basically the final assembly of those plasmids. And both of those would correspond to each other, right? There's a protocol for how do I go from the plasmids to pure, but then you can also just sort of buy the corresponding open formulation pure as well, and it all kind of works together. And yeah, this has been a sort of many years in the making, and we're quite excited about what the next year brings for being able to provide finally open formulation reagents to this community. And then finally, just a note on licensing. So everything we do is under Nucleus is open, sort of open source licensed under very permissive terms. So we make use of the CERN open hardware license. It's the permissive variant for essentially everything that I've described. The software itself has an MIT license, and we also apply like a Creative Commons license for dev notes, just because it's a very known quantity, right? But it's a little incomplete in some sense, right? Open access provides you the ability to use and reuse or use and modify figures and text from papers, but we want to make sure that people can actually practice making the systems described. And so the CERN license there gives us coverage for that. And that's that. So at this point, I guess I'll leave it to questions. And thanks for your attention.
Thank you. Thank you so much for sharing. Yes, Yankee, you have.
I think Ellen had a question in chat earlier, but I think your last couple slides might have answered that, if that's correct. I kind of wanted to know what inspired you to start all this, Anton.
Yeah, I mean, I think the sort of motivation in the earlier part of the talk, right around just this massive gap in sort of what's been demonstrated versus what uh, what can be put together, right? There's no solution for stitching together the different things that have been demonstrated in the literature. But I think at a more just basic level, um, there's if you do a PhD and you spend many years working on that sort of one component, there's a sort of sadness that comes at the end of it where you might ask yourself, what was it for? Will anyone be able to pick this up and build something, build something beyond that, right? And frequently, I think the answer is unfortunately no in a lot of the life sciences. And uh, so being able to provide a very concrete way for saying, hey, I've made a valuable contribution, and now here's the framework that allows other people to build that up, right? And actually contribute to a practical common body of knowledge is a big motivator for me in doing all this work. And of course, I think, we've got um, many people working on this now at this point, and everyone kind of has their own motivation, but that's my reason.
Okay, thank you so much for sharing. So everyone, as always, please feel free to share your comments and questions on the chat. So Yankee just shared some interesting resources, BNEX's website, and also their white paper. You can access them on the chat. And as Yankee also mentioned, we had a question from Ellen, exactly focusing, and you ended up getting to that part on your presentation, going beyond the online resources, how you can actually access the solutions, especially on the cell-free options. I don't know if you want to comment more about how has been this physical part that I think that resonates a lot, also with the
Yeah.
work that we do at Recon.
Yeah. I think our whole philosophy around developing this is that it starts in the lab with the physical materials. So yeah, we build stuff and then we kind of build the digital tools to support it after the fact. And in terms of access to the physical materials, like I said, we have the plasmid collection that's available under OpenMTA, and then from those same plasmids, right, we're developing the sort of cytosol kits that I described earlier, and that'll be released in 2026 at some point, for at least for a broader release. And I think one of the things that we're navigating how to implement exactly right is something like, doing discounted pricing, heavily discounted pricing for people that want to use this to develop open source artifacts, right? So it's a little bit more of a flexible approach than like a copyleft licensing approach, which is a little bit more restrictive in some sense. But that's an angle to this that we're heavily exploring, and would be, request for comment on that.
Perfect. Yes. Great to hear that. Thank you again. Well, everyone, do you have again any questions, comments, thoughts, suggestions, feedback? Yes. So can you just send her question on the? Oh yes. Hi, Ellen. Do you also want to comment?
Yeah, I always I mean I think this is brilliant, and it's funny because I work in a company right now where half of us are biologists and the other half are engineers, and it's really hard to talk to each other because we don't understand each other's processes. And I think that this would go a long way in actually bridging that gap uh, between the engineers and the biologists, even better than sort of the standard synthetic biology, if you see it in terms of the way a software developer would see it.
Mm-hmm.
So I think that's brilliant. But I was wondering, in these systems, do you actually have, and this is my ignorance, do you have several artificial cells that work together in a community sometimes?
Yes, that's definitely possible. I guess I'm not quite sure where you're going with that question, but there's sort of two ways of imagining how we get more complex systems, right? One is, a small number of components in a large sort of consortium of cells, and then the other one is more and more integrated components into one cell, and it's sort of unclear which approach is useful. Maybe it's probably both, but that's definitely doable.
Can a synthetic cell feed a non-synthetic cell energy?
Uh,
I have a
yes.
specific problem that I'm interested in, and I can't
Okay.
really go any further, but
Yeah.
can it act as a source of electrons or, NADH or some other component to a living cell?
I would suspect yes. I guess it depends a little bit on the, kind of requirements, what kind of flux you need, but I'd be happy to chat offline on that.
Yeah, no,
Yeah.
I want to, but it sort of opens up this crazy possibility of all of these sort of living in quote unquote non-living interactions.
Yeah. Yeah, exactly. Yeah, both from also from a metrology point of view, right? Having kind of like sentinel cells um, is a pretty big sort of design and application space there. So would be
And
very curious to know what you have in
were
mind.
you at iGEM this year?
I wasn't. One of my colleagues was.
The
I was there
team
last
that
2 years
won
ago.
had a
Yeah.
system for linking two cells together,
Mm.
two completely different cells together. So I'm
Hmm.
imagining like a living cell with all of these synthetic cells surrounding it, all linked to it,
Yeah.
like interacting with it.
Yeah. 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 Yeah.
It's a new world.
Definitely. I see a
Exactly.
comment in the chat about distribution in the
Uh,
US. Yeah.
yes, exactly. So we've previously connected about your work, and we continue very impressed with what you've been doing.
Hmm.
And at the time we talked about the distribution of the Pure system.
Hmm.
Have you increased your customer base since then, especially because you were only distributing in the US at the time?
Yeah, we've definitely distributed beyond the US, the plasmids, and then customer base. Yes, I think probably the most concrete example there is with sort of that developer cell project I had mentioned, where we're supporting 12 different labs to build on top of uh, Nucleus cytosol, right, and build these demonstrations. Um, and I guess to Ellen's point, it's been a we ran a workshop for that, and a lot of it I think was thinking about sort of bringing the engineering mindset to the forefront, particularly since the development timelines associated with that are quite quick, and so you need to think, uh, yeah, with an engineering mindset. Um, but yeah, broad distribution of the cytosol. I think it's a little bit uh, unclear when that'll emerge, but sometime in 2026. In the meantime, we're using it primarily to support sort of existing collaborations, at least for the next probably 3 to 6 months.
We're looking forward then to your next presentation, 2026, maybe in 27. Perfect. Do we have any other questions, comments? Well, then I might have a question. So everything around our community, everyone here, we are all in favor of collaboration. And you mentioned you're always open for feedback and contributions. Do you have like a channel where anyone interested here can contribute, especially to the work with Nucleus?
Yeah, that's a great question. So I'd recommend as a starting point, we just launched our own forum, our own discourse forum. So I'll share the URL for that. I would say if you want to get involved, just sign up there and say hi. You can DM me through the forum. And yeah, open for any conversations and feedback.
Perfect, amazing. Great. Thank you so much. Well, yes, we have a thanks from Brooke. Thanks for the enlightening presentation. It is indeed a great way for us to wrap up the year with our community calls. Do you have any other comments? Oh, good. Perfect. Well, everyone, then, Yankee, you don't have our end of the year comments. As always, we want to thank you all for being with us over this year. So we had 20, we had 12 community calls, over 20 speakers, because this year we had a lot of double sessions, which were amazing. And we're very excited to have all of you engaging with us, presenting about your work, advancing open science, advancing biotechnology all over the world. So really, thank you so much for joining us. Soon share about our January community call. I know for the south side of the world will be summer and you might be enjoying a very nice holiday, but we do hope to see you all for our next community calls in the coming year. And yes, Yankee, I don't know if you also want to mention something.
Hi, hopefully my connection holds for a bit longer. So I've just posted in chats the forum link that we have about Anton's talk today. But feel free if you'd like to add any further questions, comments, and we can also certainly post Anton your forum link there as well, so that we have more cross-pollination between the two communities. But no, thank you. I just want to say thank you so much for presenting to us today. It's really nice to kind of touch base with folks from earlier on and how they've progressed now, and all the best for how BNext and Nucleus go on for next year. We'll be keen to hear more. I think, as Sibeli said, we are wrapping up for this year, but if anyone in the community who's joined us today, and thank you so much for joining old faces as well, and especially since we shifted the time a little bit today. So if you do have any comments, any highlights you want to say for what you've done this year, we've got a few minutes for you to share. And if not, we'll call it a day and wish everyone happy holidays.
Okay, we have a bit of a shy audience today, no problem. So yes, as Yankee said, thank you so much, Anton, for joining us, being part of our community for a while now. As I mentioned, we're very excited to see your next presentation and how you grow your work at BNext, Nucleus, and other projects. We have more thank yous on the chat for your presentation. And again, thank you to everyone here from our community that has been with us this year and beyond. So happy holidays for all, and whatever holidays you celebrate. Wish you all a great rest of your year. Please rest, very important. Rest, everyone. You decide. But again, thank you so much for being with us. And we do hope to see you for our January community meeting and our meetings in 2026. Thank you so much.
Thank you. Bye.